Hello everyone. Today we are going to see the transgenic animal, that is our one of the module in animal biotechnology and stem cells technology subject. So overall, transgenic animals are very much useful, beneficial in the animal sciences, in medical sciences, in healthcare, everywhere. Because as far as the we have discovered, that means humans have discovered the DNA material in 1952, and then ultimately there has been like lots of research, lots of discoveries has been made through different researchers, different laboratories till now. And so we are successful in extracting that DNA material, we are cutting that, we are pasting new genetic material inside the genetic material of any organism in cells. So these all things are possible because of genetic engineering. And so the complete if you see the history, it all the cloning began with the Cohen and Boyer, these two scientists, around in 18th decade, 1982 something. They have cloned the first microorganism, and then ultimately we were able to clone the animal cells also based on different transgenic methodologies. So today we are going to see the transgenic animals and the overall how exactly we can construct these transgenic animals. If we see the traditional mechanisms, when these scientists or different researchers, they were able to make the animals more efficient for our livestock. So like these traditional breeding methods, generally they were using some selective breeds, selective organisms, right? And so for many generations these selective matings are required to improve our livestock. So the domestic animal which are generally useful for high milk yield or meat production, wool production, even though for the like for chickens we generally use them for egg, high heavy egg production. So these all organisms that if we want to breed them very properly, and if we want a good uh, the organism with the capability of producing uh, large amount of livestock, then we used to use these traditional breeding methods. Now the combination of these mating and selection, it's kind of time consuming at that time, and it is too costly because we need to bring the quality animal or good quality animal from one place to another place. The transportation cost is also included, then again the overall labor cost is also there, right? But the technique, that technique was quite successful, but it is laborious and time consuming. However, it is an effective genetic line has been established, and so it becomes difficult to introduce new genetic trait by selective breeding methods, because if we are building a good quality animal or the livestock out of selective breeding method, then we are getting animal with the same genetic pool which where these parent contents, right, the alleles which their parent content. But if you want to introduce a specifically new kind of gene, a new selective gene which has a good trait, or even if we want to delete some the lethal genes or the disease producing or disorder producing genes, then of course this selective breeding method was not useful, or that method was not enough to give us a result. So for that purpose we were using genetic engineering. Now what is exactly methods in the genetic engineering? First of all, if you see the clone gene is very much important. A clone gene is injected into the nucleus of fertilized egg, right? So generally there are two types of methodologies, but if you see the fertilized egg, then the fertilized egg is the big in the volume, so it is easy to inject that genetic material or the clone gene into the ovum, so that it can, this is the nucleus, the genetic material, it can easily go and bind to its chromosomal material, and so we can breed that egg, or we can uh, inoculate that egg again in the foster mother of that particular species, and then we can get the transgenic animal. So this kind of methodology is actually, it is useful. Some of the offspring derived from these implanted eggs carry a clone gene, okay, in all their cells. So that kind of animals, that kind of lines of animals, we calling it as germ lines, okay, germ lines. So animals with the clone gene integrated in the germline cells are bred to establish new genetic lines, and so that's what we call it as transgenic animals. So there are so many applications. We'll come back to that after our techniques, okay, for the application. So we'll see what is the different terms that is useful in transgenic animal. During 1980s, the idea of genetically manipulating animals by introducing genes into fertilized egg was converted into reality. And so what is meant by transgenic? Transgenic means an animal whose genetic composition has been altered by addition of foreign, okay, foreign DNA or exogenous DNA or interested DNA, okay, and that is called it as transgenic. Then what is meant by transgene? Transgene is meant by the DNA material or the DNA that is actually introduced is called it as transgene. That DNA could be anything. Anything means R selective genetic material which has particular capability. Then the transgenic technology, the overall process, the inoculation of transgene, then cultivation of these organisms, and then the increasing the breeding capability and getting heavy amount of that population of that particular transgenic animal is all technologies called it as transgenic technology. Then the next thing is come up over here, that is what is meant by farming. So farming means what? Farming means the increasing the amount of this transgenic animal in a multiple amount, right? We can farm this transgenic animal for the production of livestock, okay, and that is what it's called it as farming. So basically if you see the methodology to produce particular transgenic mice, okay, so there are not different methodologies. So we'll take an example of mice. Firstly, the transgenic methodology in the mice, there are three main techniques. One is retroviral vectors. There are several kinds of retroviruses which contains RNA as a genetic material. So what happened, because of reverse transcription, they convert into DNA material and they insert that DNA material, okay, into our chromosome, into the chromosome of that organism, the host cell. And that's why these retroviruses by natural they has a capability to insert genetic material. That's why we are using retroviruses for our transgenesis, okay. The next one is microinjection. Microinjection is simply to inject particular cells with a microinjection. So whatever genetic material is there, we inoculate into the cell, into the nucleus, if you see, into the nucleus, and so ultimately that vector molecule or whatever genetic content molecule, it will go and bind to our genetic material, okay. So this is what the microinjection is. Then the next one is introduction of genetically engineered embryonic stem cells into a early stage of development embryo. So here we can see that after fertilization we collect the embryos out of it, which contains the stem cells, which has a capability of stem cells, and we can re-engineer them like with the help of microinjection, and we inoculate that stem cells back into the fetus, back into the embryo. So ultimately what, some of the germ lines or most of the germline cells, they got our transgene which is present in their chromosomes, and ultimately this organism will develop that property, okay. So firstly we'll see what is retrovirus vectors. Retrovirus vector, in this technique there is a donor female. So what do we do? we extract the ovum, or we extract the embryo, not ovum, sorry, we extract the embryo out of it, out of the donor female, by injecting heavy amount of several several HCG hormones, okay, and so we got the ample amount of embryos out of it. Once we extract the embryo, which is just fertilized, and the embryo should be having a 8 cell stages, okay, 8 cell stage embryo, where each and every cells in that embryo has a stem cell totipotent property, okay, not a pluripotent, totipotent property. After 8 cell stage, there is a formation of morula, and then formation of blastula, and then formation of gastrula. These embryonic developmental stages are there, and now in the gastrulation, 3 germ layers produces, right? Endoderm, mesoderm, and exoderm. And so these 3 layers has a pluripotency property. Now here, if we select that one of the stem cells out of it, we inoculate within that cell, we inoculate our transgene, which is inoculated in the retrovirus or in the any vector molecule, okay. So we inoculate it. There is a recombination. With the help of recombination procedure, they inoculate into the that particular stem cells. Now there are types of retroviruses, like lentivirus has that property, lentiviral transfer vector for introduction of transgene into animal cells. There is the segment of DNA, you can see that here is the one of the terminus lentivirus. Terminal segments are there, 5' and 3', where we inoculate. These are several genes, Psi gene, PPT, and promoter region, and here is our transgene, okay. And so which is inoculated or which is cloned with the help of different methodologies, and so we clone that whole vector, okay, as a virus. We inoculate that vector molecule or the retrovirus molecule into the stem cells. So stem cells get infected with the lentivirus, and ultimately what will happen, because of homologous recombination or several recombination procedure, these transgene will be inserted into our chromosome, into the chromosome of these stem cells. And so once we breed these inoculated embryos into the other female, the female, we generally we insert so many hormones, actually the what do we call, the pregnancy related hormones, like choreogonotropic hormones. We increase the capability of what do we call, the pregnancy, to get the pregnancy, and so we inoculate that particular embryos in the female, and so ultimately once these the transgenic founders, when they produce, then we'll see the characteristic. So to check that whether which organism is having our transgene, there are different methodologies are there, okay, there are different methodologies. The next, we'll see these methodologies afterwards. Now second method is the DNA microinjection methods. Now what do we do here in the DNA microinjections? It is very beautiful technique that we can select not embryo but the gamete itself. So what do we do? When there is a fertilization takes place, we immediately take the embryo, okay. So now in the embryo, the male pronucleus is in the big, okay, male pronucleus is a big in comparison with the female pronucleus. So it is very easy to see that male pronucleus or to locate that male pronucleus, and we can inject with the help of this injecting pipette, the micro injection. We can inject our vector molecule, that is transgenic molecule, whatever it is, we inoculate in the male pronucleus. So what will happen? This transgene will be inserted into the chromosome of that male pronucleus, and ultimately because of the chromosomal cell division, ultimately the embryo produces. We implant that embryo in the foster mother, into that foster mother, right, and then ultimately we can get these species. So again, in which organism the transgenic characteristic has been produced, that also we need to check, and there are different procedures for that. Okay, so just remember this is the DNA microinjection technique. Now the other technique is called it as the embryonic stem cell method. Now what is the embryonic stem cells method? Like earlierly I have said, we just select the embryo, it is kind of blastula, we isolate these inner cell mass. Now you all know that inner cell mass has a property, okay, of totipotency. So we isolate these inner cell mass, we culture them, and after culturing we inoculate our transgene with the help of two different techniques. One is our retroviral technique and the other one is microinjection also. Once we inoculate our transgene, we select that particular cell which is showing the transgenic property with the help of several selection methodology. We enrich that transfected cell, embryonic stem cells, we culture them, and ultimately we inject these cultured inner cell mass or cultured cells which is having a transgenic property again into the blastula, either same or the different blastula, recipient blastocyst, and we implant that embryo in again the other mother. Okay, and so again we'll get the progeny. So there are again methodologies, several methodologies are there where we can see which organism is having that transgene and which shows that property, is it? So this is the embryonic stem cell methodology. So guys, we have seen different methodologies of integrating DNA, like the embryonic stem cells method, then we saw DNA microinjection method, then we saw retroviral vectors method. Now we are going to see the targeting DNA vector and how to select, how to confirm our targeting DNA is integrated or not, or targeted gene is integrated or not. So for that we need to arrange a vector molecule, or we need to select a vector molecule according to our selective procedures. Suppose there is a vector component and the arrangement of gene, different types of genes, different genes, the arrangement is very much important. So first of all we should know that the thymidine kinase is an it's a kind of enzyme which has been we have isolated from the herpes virus, herpes viridi. So thymidine kinase 1 and thymidine kinase 2. So these are thymidine kinase genes right now we are using. Then second gene that is very much important is homologous binding site 1, homologous binding site 2. Okay, so these homologous sites, these sequence of DNA is as similar as our host genome or the sequence of the host chromosome. Then next gene that we put here, it is the neomycin resistant gene, then our targeted gene, and then homologous binding to site. Okay, so this is how we arrange the vector molecule. Now what happened, what is meant by selection procedure? Now see here, so after the arrangement what happened, if we transfect that vector molecule in terms of this is the vector molecule, if I join these, okay, so this is the vector molecule, and if you see this vector molecule will transfect, okay, the vector molecule will transfect the our engineered cells, that is our stem cells, right? And stem cells having a nucleus. So once it interact with the host genome, now this is the host genome guys, this is the host genome, this is okay. And what happened once it interact with the host genome, with the chromosome, okay, what will happen, it will interact somehow at any spot, it will interact ambiguously. Like that could be happen. But now here what we have did, we did some changes. We have kept some homologous binding site over here and homologous binding site over here, so there are much more chances to interact the host chromosome, the same sequence. They can, the recombination can be happen. Okay, that's what we called it as homologous recombination. Okay, so next that we need to see that what happens when the transfected cells, we receive the transfected cells. So what happened after the transcription, sorry, after the transfection, some cells which is having the neomycin resistance gene, if suppose there is an integration of neomycin resistant gene, what will happen, these neomycin resistant gene will produce a kind of cytotoxic compound, or they are resistant to the cytotoxic compound that is known as ganciclovir 418, G418. Okay, so that's why neomycin resistant gene is very important. So if we are getting these cells which is transfected now, and we are culturing them, or we are selecting them on the petri dish, so which cells we receive generally? The cells which is having a neomycin resistant genes. Okay, so now next, the transgene are incorporated into the chromosome. After the transcription, cells are selected based on resistance to both G418, okay, and the compound that is ganciclovir, okay, which becomes cytotoxic to cells that they synthesize thymidine kinase. Now here two things are there. Okay, so these are two-way selection methodology. So this is the media. Now in the media we have added G418. Okay, we have added G418 also, and plus we also add ganciclovir. Okay, it's an antiviral drug, basically the ganciclovir drug. So what happen if there is an integration of thymidine kinase gene in the host chromosome? What will happen? The host chromosome will produce TK, thymidine kinase enzyme. Okay, it will produce thymidine kinase, and what happen? This thymidine kinase will turn this ganciclovir into cytotoxic compound, and this cytotoxic compound will kill that cell. Now I guess you are getting my point. Okay, so what happen if there is only recombination on the homologous site? Then which cell will be survive on the media? Those cells which are properly integrated, the cells which the chromosome which is properly integrated with the vector chromosome, is that part, only that part. If there is insertion of any TK gene, cell will not survive. If there is an insertion from this site, okay, if there is a absence of neomycin resistance gene, of course the G418 compound will kill the cell. Okay, so this procedure is called it a. Now what is meant by positive? It based on neomycin resistant genes, it is a positive. Okay, and the based on like we can say that these kind of selection is called it as positive. Okay, next, now here in the B site there is a false site, right? Suppose there is a the recombination is not there at the particular site, so that could happen. This gene could happen where the TK gene is present, homologous binding site is also present, and neomycin resistant cells is also present. So what will happen? There will be no growth of any cell. So either cells are transfected with the gene, but still they cannot able to survive. Why? Because they contain this TK gene. Is it? So this is called it as non-specific integration. Okay, and so cell will be not, these cells will not be selected based on what? Based on these what do we call it? The non-specific, because of non-specific interaction. Okay, this is called it as based on G418, this is called as a positive selection, and based on TK gene called it as negative selection. Okay, I hope so. Now here we are going to see the testing for non-specific interaction and homologous recombination in the transfected cells with the help of PCR technique. So you all know that what is meant by PCR. PCR is just like this is the DNA fragment that we need to amplify, we inoculate it, and so for that DNA fragment, what is necessary? For the DNA fragment to amplify it, we need the primer. Okay, one is forward primer and the other one is it's a reverse primer. So these primers are very much important. So primers actually hybridizes, or it hybridizes to its exact complementary site. Okay, and so ultimately they will initiate its transcription, kind of say. Okay, the initiation of preparation of DNA molecule, and so ultimately we get millions of copies of these amplified fragment. So without primer you cannot amplify the specific fragment of DNA. Now here what happened, here if you see how exactly we need to construct the vector, this is very important. So here there is a homologous binding site is there. This is a unique segment which is compatible to one of the primer, forward otherwise reverse, basically reverse primer, which is here. Then the next one is targeted gene, that is very much important gene for us, and the next one is homologous binding site second. Now see guys, for the selection of primer region, what we actually select, the homologous binding site is, now we know that, which is exact the homologous to the chromosome region. Okay, and here the site just the chromosome, one of the aside chromosomal site is here. Now we have prepared the one of the primer from that chromosomal site, and the one primer, so I'll show it with a different color. So one site from our unique segmentation. So the integration, if there is a proper integration on the homologous site, if there is a proper integration, what will happen? There will be the amplification of the DNA molecule. But what happens here? There is a attachment of okay. So there is an attachment of this DNA, this vector molecule, to non-specific site, not that specific site. So what will happen? The primer will not bind to the transfected gene, or there will be non-specific integration, and so there will be no any amplification. So we just don't find any band on agar gel electrophoresis. Okay. And in the second procedure, there is a attachment, a proper attachment to HB1 and HB2. What will happen? There will be attachment of primer, successful attachment of primer P2 to the chromosomal site 1. Okay, and the P1 is already there because unique segment is already there. So P1 attaches over here, and the amplification of DNA material will be there, that segmentation. So we see the band, and we can confirm that there is a specific integration of our transgenic molecule, and so transgene will be able to express its characteristic in terms of protein synthesis, right? So this is one of the methods which we can do. The other one, which is again very much important, that is gene knockout and gene knock in. So what is meant by knockout? That is, knockout means just cutting it down and throwing it outside. That is meant by knockout. Okay, that generally knockout term we generally hear it from boxing, right? So knockout punch, right? So like just shut off that gene, okay, cut it and throw it outside the cells, or the discard it. So that is meant by knockout. Now here how exactly we are using these terms, that we'll see that. So gene disruption by a targeted homologous recombination is called it as basically knockout. The target vector carries the selectable marker gene. Okay, selectable marker gene. Now what is the mean by selectable marker genes? This selectable marker genes which has our transgene also, either the corrected gene also, okay, selectable marker, the corrected gene with any other antibiotic resistant gene, or we saw that positive negative selection, how exactly The thymidine kinase is helpful in selecting the transfected cells. So this selectable marker gene is that vector. Okay, so now what happened? This is the target vector. Sorry. So this is the target vector, you all know, this is it. Here it is the chromosome site. The first one is the chromosome. There are segments of different genes are there, 1 2 3 5 5. And so what happened now, there is a these 2 3 5 genes are, I guess, these genes are defective genes, suppose. Okay, imagine these 2 3 4 are defective genes, and we need to take out these genes properly, because these are making that animal it will leads to any disorder, or might be these 2 3 and 4 numbered genes are responsible for any kind of certain disease. So we need to replace it with the other corrected one, or either completely intron, which is not expressible at all. So for that purpose, what do? We generally there will be homologous recombination. Here homologous sites are there. There will be recombination with the target vector with the chromosome molecule. Okay, and so what happened, we'll receive, this is the promoter region which is attached to homologous binding site 1 of the homologous binding site, promoter region, gene number 1, then our selectable marker gene, gene number 5, and the polyadenylation means termination of that the gene molecule particularly. So that kind of property, that is both, this is called it as knockout, because we are disrupting this gene material. Okay, we are taking out, and in other way we are adding new gene also by discarding the existed genes. What we are adding, we are adding selectable marker gene instead of that 2 3 and 4 number gene. So that process is called it as knock-in. Okay, I hope so you got this one. The another system which is again very important, that is modification with the gene, because earlier what happened with the other technique, you cannot control, the regulation is very much important. So it should be like a switch, you know that in our home there is a switch for any light or any electric gadget, right? If we switch off the button, then immediately the that electronic gadget just shut off. Okay, and if you switch on the working of that electronic machinery is on. So that switching is very much important, and that we have used one of the fantastic technique which is already happening in another bacterial system or bacteriophage system, and we just took it for our usage, right? Genetic modification with Cre-LoxP recombination system. So now what is that Cre-LoxP recombination system? It is the process that selectively regulates the expression of genes within a specific somatic tissue or cell type. Okay, now here the bacteriophage P1 is one of the several type of viruses that infect E. coli bacteria. Okay, bacteriophage P1. So that bacteriophage P1 is having a double stranded as a genome. It is about 100 kb in a length. What happened during the infection, when these bacteriophages infect the E. coli cells, the linear P1 genome forms a circle. Okay, and that forms a circle. This circularized P1 DNA act as a template for the replication. Okay, replication of that viral DNA material. So the template which it is used for the replication for the production of viral genome during the lytic cycle. So you all know that bacteriophage has both cycle, lysogenic and lytic. Okay, I'm not going to tell you the detailed story of these two cycles, but here lytic cycle, what happens in the lytic cycle? So what happens on the rare occasion, this circularized P1 genome actually it integrates into the E. coli chromosome, and so what? The circularization and integration of P1 genome are mediated through a specific product of gene, and that gene is called it as Cre gene. Now what is the long form of that Cre gene? The long form is circularization recombination protein, or circularization recombination gene. So this protein is also called it as Cre recombinase. Now you should remember CRE recombinase. Some says that Cre, some pronounces it as CRE. Okay, so it specifically, that Cre recombinase is specifically cleaves that DNA the P1 genome or P1 DNA, and again it helps to recombine DNA material, again self recombination, at the particular LoxP site. Now what is LoxP? It is the site, okay, where, which is the locus of crossing over, okay, in P1 genome. So that is called it as locus of crossing over X. So that's why it is called it as LoxP site. Now I know that it is bit of confusion because of theory. We'll just shift to the diagram what happened. So this is the LoxP site, guys. This LoxP site consists of 13 base pair, okay, it consists of 13 base pair. These are inverted repeats, right? Inverted repeat. Now what happened? A T A A C T T, okay, C G T A T A. So this is the 13 base pair, and again if you see there is a repetition inversely, okay, inverted repeats, A T A A C T T T C T A T A. I hope so you are getting my point. What it is, inversely repetition, okay, here also, and this repeat is very much important. Now what is the in between this 13 base pair repeating? There is a 8 base pair separator or 8 base pair spacer is there, C G C A T A C A T, okay, again. So this spacer is again very important for the cleavage. It is helpful in the cleavage side. So this is actually called it as LoxP site. Now what happens to this LoxP site? So this Cre-LoxP recombination is actually coming together of two remote LoxP sites. What happened in the bacteria? Let me show you what happened into the bacteria. If there is this is the E. coli bacteria, right? And this is the I'm not very good at diagram. This is the bacteriophage, and it is integrating its DNA material. It is now circularizing, right? Now if we enlarge one of the fragment, okay, if we enlarge here, now what happened? See, there is a Cre protein, okay, this is the Cre protein, okay, and this is a LoxP site, one of the LoxP site, okay. There are several addition of any other genes are there in between, okay. This is circularizing DNA, and again there is a LoxP. Now what happened? This Cre protein, it synthesizes its Cre recombinase protein. It will go and bind to first LoxP site, okay. It will recombine, rotate the gene, and these L1, okay, these L1 will go and bind to previous LoxP site. So 2L excuse me, 2L Cre LoxP sites will recombine over here, okay. 2 LoxP sites are recombining, and what happened? There is a cleavage. When there is a cleavage happens, what will happen? Suppose this is the next DNA material. So what will happen? This circularized DNA molecule, it will shift and it will form with another completely separate DNA material, which is knockouted, or it is been removed from the sequence. And what will happen? What will happen? A new type of gene will form. So again, Cre recombinase protein, okay, the gene, that L site, and the next gene, okay. So this is the gene which is extracted from the sequence, this red one if you see, and the new site, the further that sequence which is now attached to this L1. So I guess, I hope you are, this is the 1L site which is still over here. So I hope so you understood the exact phenomena what happening, okay. So we'll see that in the one of the figure. So these Cre recombination, it actually coming together in the two remote LoxP sites. So two remote LoxP sites, I hope so you understood what is meant by two sites, each of which has a 2 bound Cre recombinase molecule. The cleavage by the Cre recombinase within the spacer region between the repeat sequences, and what will happen in the exchange of joining the DNA strand to form the recombined DNA molecule. So this Cre-LoxP recombination system can function when the LoxP sites are widely separated. Now see what else exactly happens in the diagram. So what happened? The outcome of Cre recombination, so first of all we'll see the outcome of Cre recombination. It depends on orientation of LoxP site, guys. Now in the figure A, see in the figure A, the orientation, what will happen? This is the orientation, okay. This is like say 5 prime to 3 prime, okay. This is the sequence. So if each and every sequence is 5 prime to 3 prime, same. So now what happen if there is a integration of LoxP site is in this orientation, suppose, and the other LoxP site in this orientation, and this is the DNA. Now what will happen? The Cre recombinant molecule will come, it cut down its LoxP site, it joins this LoxP site and cut down, and after the change, what will happen? There will be inversion. There will be inversion of the molecule. Why? Because orientation LoxP site is different. Okay, orientation is completely opposite to each other. The sequences are opposite to each other. So that's why what happened. Now see guys, this is very fantastic phenomenon is happening. So orientation of our middle segment, what was it? It is 5 prime to 3. It is completely changed and it will make reverse DNA. It will cut and change and recombine again to that DNA molecule. Okay, and it will change its orientation, its direction. Okay, sometimes it happens that, and there are many more application of inversion also. We are going to see that, but this is what happened. But in the second part, what happened if the LoxP site is oriented on a same direction, okay, forward direction, suppose, and the gene is also in the forward direction. Now what happened? The Cre protein will come. This is the Cre protein will come. It will bind over here, and there will be recombination of these two LoxP site will be there. And what will happen? As I said in the previously part, the central gene will be excised. Okay, there is an excision of gene, and only one LoxP site will remain, and there will be knockout. Okay, so this is what happened. Now see, we'll take one example. Now guys, this is very important. Now see, this is a mouse, suppose. Okay, here I'll just show you. This is the one of the mouse. Sorry, my drawing is not. Okay. This is one of the mouse, suppose, I guess. And now this mouse we have inoculated, okay, transgene with the help of different vector methodology or embryonic stem cell. We have inoculated a Cre gene, okay, Cre gene over here, CRE gene. And now that transgenic mice we have selected based on different PCR method or positive negative selection, and now we got the gene, completely transgenic line of this Cre gene. Right? Oh, now we know that one species having a Cre gene, and suppose this species is a male species. Take example, male species. Now there is another species with the, this is the mice, right, with the any other gene which contains LoxP site. Okay, the gene contain LoxP site. Now here, see. Now what happened here? This is the first animal, and this is the second animal, contains a strong promoter, the exon 1 LoxP site, exon 2 LoxP site, exon 3. Suppose this is the corrected gene, and now we are preparing the diseased animal. Now what is the diseased animal? Particularly exon 2 is responsible. Exon 2 is responsible for any specific disease, right? But now currently that organism is okay. If we excise this exon 2, then there will be disease will produce in that organism. Is it? I guess I hope so you are understanding this one. If there is an absence of this exon 2, then there will be the diseased animal. Currently exon 2 is present. So exon 2 is present, so no disease at all. Okay, no disease at all. Now this is the male, and suppose this is the female. Now if we make a mating in between these species, two species, one is having a Cre and one is having the other gene, what will happen? The progeny will generate with what? There will be recombination, there will be production of Cre enzyme also, and there will be production of every gene also. And in one of the species, one of the progeny of that two parents, the one species will be like what? The one species having both genes. The one which is producing Cre gene, Cre protein will attach to this LoxP site, two LoxP site over here. Can you see that? And there will be what? There will be excision of this exon 2 molecule. And what will be the product? The one product is degraded exon 2 molecule, okay, and the one product is remaining genome, that is promoter exon 1, LoxP site exon 3. I hope so you are getting it. Okay, and so what? These organism, these progeny will be diseased progeny. That will be the animal model for any kind of disease, right? So this is conditional regulation. I want to say conditional regulation means what? There are conditions, the both mice are healthy, but if you made them, and there will be the recombination of these alleles, different alleles or different genes within the progeny, and some progeny shows that particular disorder. So we can work on that based on our pharmaceutical any other industry, right? So there is another recombination system is also there, that is what we call it as activating the transgene. What are we activating basically? Now see, suppose, just a minute, what we are activating. Yeah, so suppose there is a termination sequence. There is a termination sequence. Suppose what termination sequence is there? Suppose here the one species is the male species or female, anyone which contains Cre gene. The female one contains promoter region exon 1, but on the exon 2 is specifically for termination sequence. That means this is the complete gene. If we transcribe and translate that gene, it will produce the complete protein molecule, whatever it is. But now see, if we cleave that particular exon molecule, what will happen? There will be production of these gene line, promoter exon 1, exon 3, and now it is inactivated gene, right? And so that's why it is called it as the activating transgene. We can activate and deactivate gene line with the help of these Cre-Lox recombination system. I hope so you are getting my point. Now next part is that we need to see is the RNA interference. Now what is meant by RNA interference? So basically there are two main methods to silence the gene expression. So RNA interference means what? We are silencing the gene. Okay, we are stopping the gene expression in the animal cells for or to study any biological processes, whether it is metabolic one, whether it is disease related one, whether it is immune system, immune response related one, anyone. So one method abolishes expression of genes by the targeted disruption through homologous recombination. Okay, the one method is called it as we are disrupting the expression of genes by targeted disruption through homologous recombination in eukaryotic system, that is called it as knockout method. And the other, it decreases the expression of target gene. Okay guys, remember, decreasing and abolishes, we are stopping it and we are decreasing the amount of expression. This method is called as a knockdown, and this one is called as a knockout. I hope so you are understanding this one. So now the expression of target gene, knockout method by preventing the messenger RNA translation using RNAi technology. Now let's see what is meant by RNAi. So RNAi, it's a double stranded RNA, is recognized by the ribonuclease called dicer. Dicer proteins are there, and dicer particularly recognizes this double stranded RNA molecule, RNAi, okay, that cleaves the RNA into smaller double stranded RNA molecule, and this smaller double stranded RNA molecule is known as small interfering RNA, that is called as siRNA. Remember this word. So double stranded RNA molecule is recognized by the one of the type of RNase enzyme, that RNase enzyme called it as dicer, and this dicer they cleaves that double stranded RNA into small double stranded RNA molecule, and this small interfering molecule is called it as siRNA. The large nucleus complex called RISC, okay, किंवा असंही म्हणू शकतो आपण की RISC. So this RISC, what is it? RNA induced silencing complex. So this is the nucleus complex. What it does, it separates the strand of siRNA and the single stranded RNA product, okay, together with the RISC and bind to homologous sequences on mRNA molecule. The nucleus component of RISC, the nucleus component of these RNA induced silencing complex molecules, then degrade the mRNA. So what these RISC does, these are RNA induced silencing complex, they degrade the formed mRNA, which prevent the encoded protein from being synthesized, okay. The short endogenous RNA, now microRNAs, transcribed from the regions of genomes of animal or any plant, are also recognized by dicer or the RISC and block the translation. So there are two things are there. One is dicer is also cleaving the RNA, and here the RISC complex, that is a large nucleus complex is there, RISC, RNA inducing silencing complex, that is also making silence, that is also disrupting the translation of mRNA. So now what happens to create these transgenic mice? To create these transgenic mice with the reduced expression of transgene, with the reduced expression of target gene, a small region of target sequence is cloned, okay, small region of targeted sequence is cloned into vector as an inverted repeat separated by short spacer region, okay. The RNA transcript, now the RNA transcript that is produced from these sequences forms the short of like hairpin RNA molecule. That's what happened in RNAi, and that is called a small hairpin RNA molecule, that is a shRNA molecule. Due to intramolecular base pairing, they actually form the hairpin like structure, okay. So shorter sequence, so specifically we are adding this shRNA which produces hairpin like RNA molecule. So short sequences are used to avoid the general downregulation of translation that is often elicited with the longer sequences. Now what happened? Shorter sequences are used to avoid general downregulation of the translation, okay. So the construct is introduced into mouse. These all construct is introduced into mouse embryonic stem cells by pronuclear injection, that is microinjection, and lentiviruses vector we generally use. The stable mouse line have been generated that produce shRNA that is processed by the animal dicers or the RISC protein into siRNA to reduce the expression of transgene. So this is what actually happens. Now this is the animal which is contains shRNA gene also. This is the promoter, okay, this is the another promoter for GFP. Now what happen if there is a if there is a shRNA is inoculated, what will happen after the integration into the host organism? This shRNA will be produced, the transcription will be there, mRNA will be produced. Now that mRNA will no longer stay in a single stranded mode. Immediately, because of internal base pairing, it will form a hairpin like a structure. And so what happened, once it formed the hairpin like structure, immediately dicer molecule comes up. Dicer has that property. What dicers do? Dicers cleave double stranded RNA molecule into short stretches to disrupt that double stranded makeup, because double stranded RNA molecule will not be able to go ahead with the translation mechanism, right? So we need to cleave that at all, because ultimately what will happen, there will be the segregation of this double stranded RNA molecule in our cells, and that will be harmful to the cell. So that's why dicer is by natural presence, and dicer cleaves these Si into siRNA, and what the another enzymes comes up, that is risk molecule, who silence or who cut down these double stranded siRNA into cleavages or the formation of translation, and this is called as gene knockdown. So at the same phase, how to confirm this is happening or not? We also add GFP protein, GFP gene with the shRNA. What will happen? GFP will transcribe GFP mRNA, and GFP mRNA after translation it will produce GFP, green fluorescent protein. And what will happen? There will be fluorescence. So if there is a fluorescence in the cells of these transgenic animal, we can confirm that there is a down regulation of gene is there. I hope so you understood this one. Now, what is meant by conditional knockdown, conditional regulation of knockdown of target gene expression using this RNAi methodology? A blocking sequences, okay, the blocker will be cloned in between the LoxP site to same oriented LoxP site. See here guys, now what happened? We are cloned our blocker sequences over here on the LoxP site. Okay, now flanked by LoxP site in the same orientation, and it is inserted in between the sequence encoding the shRNA. shRNA is also encoded, see here. Now the promoter that controls the expression of hairpin RNA, okay, hairpin RNA which has been synthesized from the shRNA, small hairpin like RNA. The Cre-recombinase mediated recombination. Now what happened here? The DNA molecule is there, and now we have integrated it. Now because of this blocker, whatever promoter is there, the expression is blocked. Okay, so expression is blocked mean what? The expression of that protein is completely blocked. Now if we suppose the expression of shRNA is blocked means what? Whatever gene is there, whatever the either the gene is present over here, see here, either the gene is here or somewhere in between this shRNA or before the block. Because of blocker, the gene will not produce shRNA. So what will happen? The other genes will produce its protein, and there will be activated organism which produces that particular protein molecule. But see what happened if we add if we mate that particular animal with the other one which is having a Cre gene. What will happen? The Cre molecule will bind to LoxP, it will cleave that blocker, it will knock out that blocker, and shRNA will produce shRNA. Transcription will be takes place because no blocker is there. It will cleaved by dicer molecule, and then ultimately with the risk molecule gene knockdown is there. So these molecules will not be produced any further. So this is what we are calling it as gene silencing. We are silencing, conditional silencing. Okay, now other technique that we need to see, that is CRISPR-Cas9 technique. Now what is CRISPR? Last year only that technique, who discovered that technique, Dr. Doudna and the Charpentier, these two the great ladies, they discovered this technique, and currently we also know that they have a lots of application in the current science. Might be because of CRISPR-Cas9 will be able to face new era in the biotechnology, in the genetic engineering, because as far as we are using endonucleases, different restriction endonucleases, they cleave molecule not that much precise. Of course they are specific to the sequences, but not as precise as CRISPR-Cas9. Now what is happened? Three things that we need to, we will see the method first of all. The three things where you need to remember: one is CRISPR, the other is Cas9, and the other one is guide RNA. So what is CRISPR? It is a clustered, regularly interspaced short palindromic repeats. Okay, this is the long form of CRISPR. And this CRISPR of a genetic information that some bacterial species uses, this CRISPR as a part of antiviral system. Okay, so what happens when any bacteriophage gets or bacteriophage makes attack on the bacteria. So bacteria uses these CRISPRs to recognize that foreign RNA molecule. They remember that RNA molecule as like the immune responses or the memory. So what memory they remember? They remember the guide RNA, the type of RNA molecule that binds to Cas9 product or Cas9 protein and specifically and cleaves that RNA molecule. So Cas9 is the CRISPR associated endonuclease molecule. It is the endonucleases that cleaves that RNA molecule. So Cas9, the CRISPR associated endonuclease or enzyme that acts as a molecular scissor to cut DNA at the location specified by guide RNA. So guide RNA is acting as a mRNA. So bacteria restores these these RNA, the RNA, the fragment which has been cleaved earlierly because of their defensive mechanism. Whenever these viral attacks is there on the bacteria, they just store kind of guide in terms of kind of guide RNA, so they will remember in for future infection of the same viruses. So this defensive mechanism of bacteria, we are using that mechanism for our purpose to cut down on the space. So how does it work? So guide RNA is designed to find and bind to specific sequences in the DNA. The guide RNA has a RNA bases, right, that are complementary to those of the target DNA sequences in the genome. This means that at least in the theory, the guide RNA will only bind to the targeted sequences only. So no any other region with very precision. So the precision is there, very much precision. If suppose this is the DNA molecule and with a high precision, these guide RNA will bind over here with a very high precision, means each and every base pair is very important for attachment. Okay, now the Cas9 follows that guide RNA to the same location. Now Cas9 molecule comes up, this is the Cas9 molecule will comes up, it will bind to that, it will follow the red one, right, the guide RNA. It will bind to that molecule DNA sequences and makes a cut across both strands of DNA. At this stage, the cell recognizes that the DNA is damaged and tries to repair it. At this stage, the DNA is damaged, no? So cell tries to repair it, but Cas9 don't allow to repair it. So it will rejoin the DNA, whatever disrupting it. So scientists can use the DNA repairing machinery to introduce changes to one or more genes in the genome of the cell of that particular our interest. So now that's what happened. Now what happened? The target sequence is there, and this is the guide RNA, right? You can see that it looks like this one, the guide RNA. So guide RNA actually attaches to the targeted site. Okay, what will happen? Cas9 will come, it will recognize that guide RNA molecule and it will cleave the DNA strand. It will not cleave the guide RNA. Remember, the Cas9 molecule cleaves the DNA material which is attached to that guide RNA. There will be the mutation or there will be the repairing mechanism, and ultimately there is a cutting down. Okay, so this is what happened. And now I want to tell you one more thing. Currently the corona pandemic is going on, and now there is a new CRISPR-Cas9 detection system now came up in the market to analyze or to check. It's as one of the diagnostic tool. The CRISPR-Cas9 technique is coming up into the market. You just see that how exactly it is working, right? Now we need to see a different model. So first of all, we need to see a transgenic model as Alzheimer's disease. It's a neurodegenerative disorder, and how we are preparing transgenic mice with the help of transgenic disease model. So first of all, we need to understand that Alzheimer's disease. What happens in the Alzheimer's disease? So Alzheimer's disease is because of neurofibrillary tangles. The production of these neurofibrillary tangles which accumulates within the cellular body of neurons. You can see here, these fragmented bodies are here. They are accumulated in the dendrons and axons also, neurofibrillary tangles. And this is because of this, these neuron cells will not able to pass any message from one end to other end. And so this is happens in the basically these neurofibrillary tangles, the extracellular aggregates are there, and the ultimately what there will be neurocortex damage in the neurocortex and hippocampus region of the brain in the patient, and so ultimately it will lead to Alzheimer. The core of these bodies, when it come up together, we can see that there is a formation of senile plaques. Okay, these senile plaques are there, and this senile plaques is composed of closely packed fibrillary structures that traditionally has been called the amyloid body. Okay, the originally amyloid bodies were thought to be made up of carbohydrates. Previously researchers was thinking that it is made up of carbohydrates, but more definitive analysis established that they are protein aggregates. So however, despite of these misnomer, the term amyloid has been retained. Amyloid, the amyloid is kind of like protein bodies, kind of say. So the what is happening, the principal protein which is responsible for these amyloid bodies is 4 kilodalton protein, called it as A beta, that is amyloid beta. Okay, or in other one we can say that beta slash A4 gene also there. So amyloid beta gene, or it is a protein, it ranges from 39 amino acid to 42 amino acid residue. Okay, and now what happened, these amyloid beta 40 and amyloid beta 42 forms are these two forms are the main variants of these two particular protein. So these amyloid beta proteins are derived from the internal proteolytic cleavage. Okay, amyloid beta protein is very much important, but how exactly they form? The internal proteolytic cleavage of beta amyloid precursor protein. So there is a production of beta amyloid precursor protein, APP, and APP degrades and form amyloid beta proteins. What happened now? This amyloid beta protein is very helpful. It is by naturally it is synthesizing. But now what happened if there is a, if this is the gene, just a minute, if the big gene APP is the big gene, and if there is a like we can say this gene is, there is a improper cleavages, there will be like improper cleavages at any site, like the cleavages should like that. Okay, the proper cleavages. But if suppose imagine the cleavages are in improper cleavages, what will happen? Because of this improper cleavage, the protein will be changed, there will be change in the structure and ultimately in the function of protein. So faulty cleavage of APP protein causes the production of amyloid beta 14, amyloid beta 42, and inefficient clearance of variants likely leads to their accumulation. And these accumulation we generally call it as these amyloid bodies. Now this APP gene is very much important, guys. APP, if there is a mutation in the APP gene, there will be faulty cleavages. Okay, so we need to find out this APP mutation and we need to replace it with the correct APP gene. Now what is happening in the animal? What we are actually doing, we are doing the animal, we are introducing APP, corrected APP gene, we are checking whether there is a application, we are making a mutation, we are making a knockout of this true APP gene, we are producing Alzheimer transgenic mice, right? Alzheimer transgenic mice, and we can check different drugs on it, different chemosynthetic drugs, natural drugs, whether they are able to cure that Alzheimer disease or not. So this is one of the neurodegenerative disorder diseases. I guess for this lecture this amount of information will be enough. For the next lecture we'll see in the next lecture, next module we'll see in the next lecture. Okay, bye bye.